about upcoming material and how it's going to go down and all that good stuff. I feel like we're on the home stretch, and I think that because, and I think I mentioned this to you in an announcement, but in terms of difficulty, we're on the downswing. That last chapter, that last set of chapters, to me, by far is the most difficult material we tackle. Even if you are in A&P2 right now, you've had immunity. You know what T cells do. You know what B cells do. Did you feel like the innate stuff I had you do was familiar? You did it, right? Say yes. Yes. All right. So, because I really want to move quickly through the easy stuff so that we can spend more time on the harder stuff. But the thing is, in A&P, you have one chapter with all of that immune stuff. Now, that single chapter covers what micro takes 4 to do. So our next exam encompasses innate immunity. That's one micro chapter. Adaptive is another. Then you have analysis of the immune response and how you do it in the lab and all that, which is a little bit new, but it's not really difficult. And some of it you've seen before because we've gone through a lot of those lab tests. So I feel like we're kind of on the home stretch, and I think you guys are mostly set up to do very, very well. Questions about any of that? I really wanted to have that third exam sooner, but I just don't see how we're going to be able to work that in before spring break, especially with the day that I missed that was unexpected. I really feel like we need to go ahead and do that after. You have a disease notebook that I want you to start working on soon. That's not something that I'm going to take from you, but it is something that's going to help you down the road. The guidelines are posted, and it's going to help us get through those last chapters without having a dump at the end. Have you had a semester like that where you get to the last week and your instructor says, well, we didn't get all this stuff covered, but here's a packet of 50 things, and I want you to learn this by the end of the term. I don't play that way. I don't, that's just not the way we do things. So as we get closer to those disease chapters, I'm going to post a list of the diseases we're going to cover. There's no way we can get to all of them in this chapter. We just can't. So I'm going to pick and choose the ones that are relevant and important, and those are the ones that I want you to focus on. That notebook is what you make of it. You can copy and paste from online, but if you do that, how much of it do you think you're going to remember? 0. Yeah, so it's better to do it kind of handwritten or typed. I prefer to type, but the idea is that you're processing information. So more details to come with that. I think that's all the housekeeping. Who's coming tomorrow? Gosh, like everybody. Good deal. Do y'all know where to go? Go to the D building. This North Metro is tomorrow. Don't go to Jasper. I had somebody ask me if they could take their poster to which Piedmont hospital. We ain't going to the hospital. It's Piedmont Park. Oh my God. Maybe you should just say it. We're going to be in Building D. So if you go into the main campus of North Metro, big Chattahoochee Tech sign, Building D is on the left. I'll have a sign-in sheet for you. I'll have a T-shirt for you. So yeah, if you already have a T-shirt, feel free to wear it. But it's a Science Outreach Volunteer T-shirt, and they're nice. Chick-fil-A will be there. So bring $10 for lunch. That's going to be kind of a big thing that we're doing this time around as well. All right. Yes. No, we'll have 2 tests in the final exam. We are. Don't worry. It's all good. I got this. You done this before. Okay. Yeah. Good. Okay. It's because most of it's geared toward that notebook stuff, but I'll load them up for you. And if you do decide you want to start that notebook, I highly encourage you to use the PowerPoint notes, which are apparently not yet posted. Thank you. But I'll get them posted. The reason I say to use the PowerPoint notes is because if you start looking up things like hepatitis online, you are going to find more information than you know what to do with. we are scratching the surface of that stuff. We just don't have time to get into that kind of depth. But this is a notebook you'll use when you take pharmacology. I got an email from a student in nursing school at Mercer who said she's still using that notebook right now. So it's going to be a good resource for you to have. All right. Okay, so let's blaze a trail through innate immunity. I tend to do this as a concept map, and I know I gave y'all. Do y'all like the fillable outlines for note taking? I think it gives you a little bit of a focus so that you're not just flying blind. So, what are our two branches for immunity? We've got innate and we've got no markers. So we've got innate and we've got adaptive, right? And so under innate, which also means what? Yeah, non-specific, which means what? You are born with it. Yeah, you're not targeting a particular pathogen. This is not, and y'all forgive me, but all my immune system analogies tend to be a little militaristic. That's just the way it is. In innate defense, you're just opening fire on everybody. Adaptive defense is an assassin. You have a target that you are going after. So those are the two major differences. And I divvy things up a little differently than the textbook does. I like to separate my branches of innate immunity into physical barriers, chemical barriers, cellular barriers, and then there's the complement system. And so hopefully your note guide kind of got you through some of that. So when we talk about physical barriers, that's not good. All right. What's your biggest physical barrier that you got? Skin. Your skin and integument. What makes it an appropriate barrier?
It's the tight junction.
Yeah, so when we're talking physical, think about the tight junctions between the cells that are keeping things from getting in or hopefully out as well. So to that end, you also have some chemical components associated with it as well. What were some of the, I'm going to put tight junctions, or TJ for tight junctions. What were some of the chemical adaptations that the skin has?
Like sebum.
Yeah. Salty. Low pH means it's acidic or basic. Acidic, good. What other chemicals we got? Defensins, dermcidin, lysozyme, all of those things I would consider to be a chemical barrier, right? And then what was the third one? Yeah. And some of these can kind of go either way. So like with cellular keratin, I mean, to me that's chemical, but it's a cellular product. Same thing with sebum. Why is sebum good for your skin?
Because it keeps it moist.
Keeps it moist and pliable so that it does not crack, because when you have a crack, you have a breach and bacteria and things can invade. All right, so you got your notes that you took in front of you? Yeah? And do you have questions about anything on there? Are we okay with these three? Because what I'd really like to do is launch into the complement system. I feel like this kind of stuff is a bit of a review, and y'all should be well up to speed with that. Are you? Is there anything there that you need clarification on before we move forward? Yes?
What about, I saw it in the chemical defenses, sebum?
Sebum?
Yeah. Is that like in the skin or?
Well, it's produced by, it's the oil that's produced by the sebaceous glands.
Okay.
And that's what we were just talking about with the pliability and waterproofing and keeping the skin intact so that it doesn't you don't get abrasion.
Okay.
Because dry skin cracks. Yeah. It's a defensive chemical. It's a natural antimicrobial. It's a destructive enzyme, and they all kind of do the same thing. So Jennifer asked, what does lysozyme do? What's the difference between that, dermcidin, and defensins? associated all of them with antimicrobials. You don't have to know a specific action for each. Does that make sense? Okay. Where do you tend to find lysozyme?
Your tears.
Yeah, tears, tarsal glands in the eyes, and so forth. Mucous membranes. What kind of immunoglobulin would you find in there? Good. IgA, because IgA always has what word in front of it? Secretory. If you don't know that yet, that's okay if you haven't done it in here yet. All right, so we're good with this. What about endothelins or endothelia? Tell me about endothelium in the blood vessels. It lines the insides of the blood vessels. We've got more tight junctions there, so more physical barrier. Blood-brain barrier in particular is associated with that. Endothelins are chemicals that are released when vessels are damaged. So they can be, and I think we talked about this in the cellular chapter as well, they can be used as an indicator of things like heart disease, vascular damage, because they are not supposed to be circulating. They're supposed to be hanging out on those vessel walls. The other thing about endothelia is when, remember the clotting cascade and the intrinsic and extrinsic, when those insides of the vessels are exposed to the outside tissues, that triggers the inflammatory response also. In other words, tissues that aren't supposed to be in contact with each other. That's what that's about. All right, mechanical, are we okay with that? This is to me less of a thing, like the flushing of the digestive system, the flushing of the eyes, the flushing with the kidneys and so forth. It has an immune role, but generally bacteria do not breach into the urinary system, which is why people who survive earthquakes and whatever can do so by drinking their own urine and sterile. It wouldn't be my first choice of a beverage, but you know, but you get the idea. That is a place that is supposed to be impervious to bacteria and other microbes. How about the normal defenses or normal microbiota?
How many the other stuff from growing?
Yeah, basically, how many species of bacteria are you home to?
Several.
A whole bunch, several, a bazillion, right? More than you can count. Most of them are beneficial. Some of them are necessary. If you don't have E. coli in your digestive tract, you're not going to be able to digest certain foodstuffs. So when you have an issue, you take, let's say you take a bunch of broad spectrum antibiotics, you disrupt the normal balance, suddenly you can have other bacteria overgrow. What's a good example of that? C. diff. Clostridium difficile normally lives in your colon, and it lives there happily because its population is kept in check by the other bacteria. When you are bombarded with antibiotics, those bacteria die off and C. diff explodes. Nasty stuff. How do you fix it? Stop taking the antibiotics, or you switch up the formula a little bit. Suppose you're in a situation where the entire gut flora is non-functioning or non-existent, then what do? Yeah, then you get something lovely, something called fecal enema. Two words that should never go together. but that's exactly what it is. They take a stool specimen from a close relative or friend. I don't know what kind of friends you gotta be, but I'm just saying. But they do, and they use sterile water and they literally infuse the digestive tract with it. It works, and it's a hell of a lot cheaper than traditional medications. It is disgusting. So tell me what the resident bacteria do for you other than helping with digestion and whatnot. They outcompete the bad stuff. They use up the nutrients. Some of them can even manufacture toxins that will deter other bacteria from growing. Sounds like you're pretty well up to speed with that. All right, we got into chemical defenses already. This business with antimicrobial peptides, this is just the generic term for what these things are. So if you've got a heading that's antimicrobial peptides, what types of things are you going to have underneath there? all the ones you already mentioned, lysozyme, defensins, bactericidin, I mean, whatever. They all essentially do the same thing. Earwax, believe it or not, is considered an antimicrobial because it is alkaline. Most of the stuff we think about acidity, earwax happens to be alkaline. All right, are we good with this? And I know I'm moving fast through this, but this is stuff you've seen before, and if you did the activity, you should be up to speed, all right. We okay so far? All right, good. This is probably where we need to start, because the complement system tends to kind of mix people up a little bit. What is it? What's up?
Oh no, I was trying to do this yesterday with the outline. I don't know where
You don't know where you got to?
Well, if I wrote this down, I wouldn't know it. So I'm hoping it was
For complement?
Yeah, well, when it goes into classical alternative.
Oh, yeah. That's why I wanted to start here. This stuff you're okay with, though.
Yeah, no, I'm always
Okay,
down.
good. Yeah, so let's start with complement. What the heck is it? Yeah, it's basically their proteins, okay? And usually they're designated with a C and a number. So if you see, C1, C4, C3, those are not Lockheed planes. Those are complement proteins, all right? So the analogy I use with them is they're like plainclothes police. They circulate all the time. They're always present, but they're not always doing anything. Something has to trigger them to get them to spring into action, in other words. And so that's where we start divvying it up as, okay, there are 3 ways to activate it, and there are 3 basic things that it does. But it's all circulating proteins, and it's considered non-specific because, again, it's indiscriminate as to which microbes it attacks. It'll go after E. coli, it'll go after Salmonella, it'll go after Clostridium, it'll go after whoever. It's not targeting a specific thing. So there's 3 ways that the complement system can be triggered, if you will. The first is the classical pathway. And what that means is that an antibody binds to an antigen. Simple as that. So that means it works in conjunction with the adaptive response. Y'all remember a little bit about B cells and how they produce antibodies? Say yes. Okay. Those are specific, but the targeting of those antibodies, or the activation of the antibodies with the antigen, triggers the innate complement system as well. You follow? Yeah? Okay. Another thing that can trigger the complement system is called the alternative pathway. I do not know what creative genius came up with this name, but really, pathway number 2. The classical pathway is antibody-antigen. Think of the alternative pathway as a series of complement proteins binding to each other. So it's a domino effect, all right? And I mean, in total, it looks like, kind of like this. You've got a C3 that binds to a C4 that recruits a something and a something else. All I want you to know is that it is a series of complement proteins binding to each other. That's the alternative pathway, okay? So that's way number 2. And then way number 3 is the lectin pathway. And it looks something like this. The lectin pathway involves a sugar called mannose, which is, it's present on and associated with our cells, but lectin doesn't normally bind to it. So think of it as kind of like you've got a weird sugar that's not normally present, and when it is able to bind to a protein called lectin, that triggers the complement cascade as well. It's not one that we see we don't deal with it quite as much as we do the other 2, so that's why we're not getting into a whole bunch of details with it. Does that make sense? So 3 ways it's activated: classical, antibody-antigen binding; alternative, complement proteins binding to each other; and then the lectin pathway is when you have lectin binding to mannose and triggering that reaction. Okay? So far, so good? Okay, so then what happens when the complement system is triggered? Well, you've got three things that can occur. You've got opsonization, which literally means make tasty. So complement proteins, it's the strawberry cover, it's the chocolate covering on the strawberry, right? So you've got complement proteins that literally bind up all the receptors on the pathogen, and that helps the phagocytic cells come and eat it. Opsonization, simple enough. The other thing that it can do is trigger inflammation. And again, that's kind of where the complement system helps bridge between adaptive and innate defenses. Inflammation is considered a defense, right? So when complement's triggered, what happens during the inflammatory process? What are the cardinal signs of inflammation?
Edema, swelling, redness.
Yeah,
Sometimes
redness,
joint
heat.
stiffness.
Yeah, redness, heat, swelling, pain. Those are the big four. Redness, heat, swelling, pain. Why do these things happen?
Because of fluid going into the interstitial space.
Yes, because when inflammation is triggered, the capillaries get leaky. They start to dilate, they start to get leaky. Why the heck do they do this?
So he can get out and fight.
They're trying to make room for the white blood cells to go into the tissues and help. White blood cells are one of the few things that can actually move out of the bloodstream. What's the name of that process?
Diapedesis.
Diapedesis or diapedesis. Good. So white blood cells can move out, but unfortunately, that means the rest of the fluid moves out too. That causes the swelling, which in turn causes the pain and the heat as well. But overall, it's considered a beneficial process and part of the immune system. The third thing that complement does is create membrane attack complexes, or MAC attack if you prefer. And so what this does is basically, you see all these little C proteins, again, these complement proteins assemble themselves into this little circular thing, and they basically punch holes in the cell membrane. A lot of holes. It's not just one. I mean, it's all over the place here, and it causes the cell to lyse. I mean, that's the thing. So if I were to ask you a question, what does the complement system do to cells, or what does a membrane attack complex do? What's the answer there? It causes it to lyse. It's not killing the cell outright, okay? In fact, there are very few immune cells. There's actually only one that can kill a cell outright, but the mechanisms are different. So membrane attack complexes, holes in the bacterial cell wall so the cell lyses. And that's really all you need to know about complement. It's complicated. I mean, the pathway and the enzymatic reactions look something like this, but unless you study immunology for research, there is no way you're ever going to have to know that. But you do need to know three ways. You need to know what the complement system is. You need to know the three ways that it's activated and the three things that it accomplishes once activated. Okay? Keep it simple. All right. So, where are we now? Cytokines. Why did we mention cytokines?
They're the signal that they send out.
Yeah, they're the voices of the cell. So anytime you're talking about an immune response or an invasion by a pathogen, you have to have communication between cells because you've got so many different branches that are relying on each other for activation. And so anything that is a signal, usually they're proteins, so they can travel from cell to cell pretty easily. They mediate. Does everybody understand what I mean when I say mediate? What does it mean to mediate something? Yeah, it means to direct. So when you hear cell-mediated response, it's a response that a cell is telling you what to do. So cytokines mediate the response by sending an alarm signal and telling complement proteins to be activated. Cytokines mediate by telling the vessels there's a problem, you need to trigger inflammation and let the white blood cells out and so forth. So there's a couple of different kind of subsets of messaging. if it's autocrine messaging, what does auto mean? Self. Yeah, so it's self-stimulation, which sounds pretty gross, but it's where Maureen thought that was really funny. Alright, so it just hit you right, didn't it? Alright, so autocrine, sometimes cells can receive the same signals they are releasing. And these signals can trigger all sorts of things, like activation of phagocytic cells, or they can trigger a cell to change its DNA, change the genes that it's expressing, change the receptors so that virus in the neighborhood can't get in, and that sort of thing. So, in addition to activating immune cells, they can have an effect on self cells as well. Paracrine signaling is happening in the same neighborhood. Endocrine signaling is across the county, right? So it has to do with distance, as all those terms refer to. All right. Other types of cytokines, I mean, they all kind of do the same thing, and I'm not going to ask you to differentiate between them because I feel like that's just a little bit too much. But I do want you to know interferon specifically. Interferon is associated with virally infected cells. And basically, y'all know what viruses do, right? What do they do? Yeah, they hijack the cell's DNA and they use the cellular equipment to start cranking out copies of viruses instead of cellular products. So what interferon does is when the cell gets attacked, it releases interferon. It tells neighboring cells, there's a problem and you need to save yourself because I'm already a goner. And it does this by causing certain genes to be expressed or not expressed. In other words, change your receptors because this virus is going to attack you. Change the receptors, the virus can't get in. Does that make sense? So interferon associated with virally
I have a
infected
question.
cells. Yeah.
Does it also give the cell that received the signal to create inactive enzymes, and so that if the virus does come over and attack, then that activates enzymes which will kill the virus?
It can. Yeah, it can. I just always associate it with, and I'm not really concerned that you're too specific with what exactly it does, just associate it with virally infected cells, because it can be a measure to determine, what you've got going on from an immune standpoint as well. All right. Other cytokines, there are a couple that you need to know on here. Histamine. What does histamine do? Yeah, so mast cells are associated with histamine release. What other white blood cells are associated with histamine? Basophils. Good, basophils. So what types of things happen when histamine gets released?
Inflammation.
Yeah, it's the typical inflammatory response. The vessels dilate, they become leaky, fluid starts to accumulate in the interstitial space, and possibly fever is activated as well. But that's what histamines do. Prostaglandins also promote inflammation. All right, prostaglandins also promote inflammation, but those are the only specific ones that you need to know. So if I showed you a picture of this cell, would you be able to put together that is releasing histamine? How? You've got granules, yeah, you've got granules, so you know if it's a cell full of granules, it's probably a basophil. But what's up here? What's C3 and C5?
Those are your complement proteins.
Those are your complement proteins. And so when you see that complement cascade, immediately start to think of things like inflammation, MAC attack, opsonization. So this is how the complement system mediates inflammation. They attach themselves to the basophils or to the mast cells, trigger the release of histamines. Okay? We all right with that? Okay. Y'all look like you're asleep. That's what I'm asking. How about white blood cells? This falls under our cellular response, right? Okay, so how are we going to remember all the white blood cells in their relative abundances.
Never let my engine blow, 60-30, go.
Everybody get that? Alright, here's how we're gonna remember. We're gonna remember, never let my engine blow
60-30.
30, 8, 2, 0. Like that? Yep. Never let my engine blow 60, 30, 8, 2, 0. And these are white blood cells, which are also known as, these are leukocytes. Leukocytes. And the reason I'm making this distinction is so you don't mix these up. Alright, so which leukocyte starts with an N? Neutrophils. Lymphocytes. Lymphocytes. Who are lymphocytes? T cells and B cells. Are all lymphocytes leukocytes?
Yes.
Yes. Are all leukocytes lymphocytes? No. And that's why I make the distinction. So lymphocytes, M is monocytes, eosinophils, basophils. So this is how you know your relative abundances. Why do we care? Y'all are happy y'all reading like, well, I don't. So, you know. Yeah, because if you know what the baseline abundances are supposed to be, and you do a blood sample from your patient, and you do a white count, have you heard of that, a differential white count? It is a tedious process. You do it by hand. You do a blood smear on a slide that's got a teeny tiny little grid on it, and you have a clicker with multiple things to click, and you count every white blood cell you see, depending upon, and you document. So if your patient has an excess of eosinophils, that clues you into what possibly is wrong with them, because each one of these does something else, does a different thing. All right, so we know our relative abundances, we know what they stand for. Now let's divvy them up into an additional group. Which ones of these are granulocytes or granular? Ben. Think Ben. Some people think about Phil as being granular, but I prefer Ben. So that's your basophils, your eosinophils, and your neutrophils. And then of course these two are agranulocytes, they're non-granular. And where are these blood cells born?
Your bone marrow?
Yeah, they all come from the bone marrow. They all start as a stem cell in the bone marrow, and then they differentiate. All right, so what are their functions? Y'all already know what they look like. The agranulocytes will start with their function. The lymphocytes are adaptive. Y'all already told me those are T cells and B cells. We'll deal with those in the next chapter. The others are monocytes, and those are the big eaters. They leave the bloodstream to become macrophages, phagocytic cells whose job it is to eat and destroy whatever pathogens they find. And then that leads us to the other ones, the mast cells, and are the granular ones. Ben, we already said. What are the neutrophils for?
They're like the first responder.
Yeah, I want you to think about it, and that for some reason that's not really, it kind of is. Think of the neutrophils as the first responders, okay, which makes sense because they're already the most abundant, right? So they're the first responders. They tend to be phagocytic as well. So they're going to go and they're going to detect the pathogens and they're going to be consuming them. They are capable of diapetesis as well. So they leave the bloodstream and head into the tissues. Basophils are associated with allergies and mast cells. So basophils, I want you to think of those as histamine. They are small sacs of histamines, and they play a role in allergic response. Okay. How about eosinophils? Yeah, parasitic worms. That's still a thing. And you know, when these defenses evolved a million years ago, that was much more of a risk than it is now. But eosinophils also play a role in allergies, but they're associated with helminth infection and things like that. So if you've got a patient who's chronically runny nose and all this business and so on and so forth, and you find that they have an abundance of basophils, what are you gonna suspect is really their problem?
An allergic reaction.
It's probably an allergic reaction. See how that factors in? So this all falls under that cellular response. Also, kind of included in this innate cellular response are natural killer cells. And there are some texts that put these with the adaptive response because they do have a targeted attack, but they are not targeting a particular pathogen. So I want you to associate natural killer cells with tumor cells and also virally infected cells. So the difference is in how they are determining who's who. So let's see how much you remember. Who knows what MHC stands for? It stands for Major Histocompatibility Complex. Think about those words, they sound big and scary. We don't like a bunch of syllables in camp, right? We got histo, which means tissue, compatibility, which means compatibility. Can it go together? So these are proteins that are present on all of your cells. Think of them as self-markers. They were discovered when people started investigating organ transplantation because they couldn't figure out why can't I just take this kidney from this person and put it in this other person? What they discovered was those self-identifying proteins or self-markers, if they didn't match up, what does the immune system do? They throw a bomb at it, right? So that's what histocompatibility means. Now, every one of your cells has, which one? Good. Every one of your cells has an MHC1 complex. Every one except who? Red blood cells do not. Why? How much endoplasmic reticulum to build proteins does a red blood cell have? No, they've got nothing. They're sacks of hemoglobin. They're not making proteins or anything else. So they are the exception. They lack any MHC. But all other cells have this MHC1 complex. What kind of molecule is it? Protein. It's a protein, so the instructions for building it are in the DNA. You see where I'm going with this? Okay, so if that's the case, what happens to the MHC complex if a cell is a mutated cell, like a tumor cell? It ain't gonna match anymore, right? That's what lets the natural killer cells see who's who. So same thing with a virally infected cell. You start expressing different proteins, suddenly the MHC doesn't look right anymore, the natural killer cell targets that cell. Do you see why that is considered innate and not specific? It's going after damaged cells of yours, but not targeting a pathogen specifically. Make sense?